Good afternoon, everyone. It's a pleasure to see you and some familiar faces and boxes and names. It really is heartwarming to see what a community we're building in the Aufzien Family Center for the Prevention and Treatment of Parkinson's Disease. This is a particularly special lecture series that's hosted by Professor Nir Giladi and I, with a lot of help from Rinat, because we're going to have showing our youngest stars. Those of you who are speaking today were awarded competitive fellowships from the Family Center, and you've been working, some of you for 6 months, some of you for a year or two, various different stages of your scientific careers. But what you have in common is you're all working towards understanding the molecular and other mechanisms with ideas of where it could be going into translation, specifically for Parkinson's disease. And I'm confident, having seen your proposals and the updates that you're written, that the work that you're doing is going to be the future, and I think can keep patients and their families very hopeful of where the future is going for them. So again, thank you for joining us. Those of you who are our guests, those of you who are the mentors of these really terrific students and postdocs, and then of course, our speakers today, those who have been awarded the scholarship. So we're going to start with our first speaker, who is Shai Herman, and she's going to talk to us about Parkinson's disease patients' CSF-derived extracellular vesicles. And please, each one of you, when you speak, let us know what lab you're coming from, and so we can hear a little bit more about where you are. So Shai, it's all yours.
Thank you, and sorry again. So hello, everyone. My name is Shai Herman, and I'm a PhD candidate in Professor Zdani's Offen Lab at Tel Aviv University. And I will be presenting my current research in the field of prion-like transmission of pathologic protein aggregates by extracellular vesicles. So Parkinson's disease is characterized by motor and non-motor symptoms, which are caused by a progressive loss of the dopaminergic neurons projecting from the substantia nigra to the striatum. This dopaminergic neurons degeneration is caused by a pathological accumulation of the neural express protein alpha-synuclein into Lewy bodies, which are basically the hallmark of Parkinson's. Barak and colleagues postulated what they call the dual heat hypothesis. After they observed postmortem human brains, they noticed that the Lewy body pathology actually develops first in the olfactory bulb and also in the gastrointestinal tract. This hypothesis also correlates with symptoms which we see in patients year before the motor symptoms begin, like olfactory dysfunction, REM sleep behavior disorder, and constipation. Only years later, during disease progression, that we actually see the Lewy body pathology is observed in the midbrain, affecting the substantia nigra and other regions. So like we said earlier, Parkinson's disease is a progressive disease. And this is due to the fact that alpha-synuclein can pass from cell to cell in a prion-like manner. It means that during the pathogenic process, alpha-synuclein misfolds, it forms toxic oligomers and insoluble fibrils. And these insoluble fibrils and oligomers can pass from cell to cell in a prion-like manner. So misfolded alpha-synuclein can pass from a healthy cell, induce the native alpha-synuclein in this healthy cell to also adopt the irregular conformation, and eventually aggregate and lead to cell death. In recent years, much evidence indicate that nanovesicles, called extracellular vesicles, or eevees, also sometimes known as exosomes, play an essential role in the propagation of pathological alpha-synuclein. So eevees are released by all cell types. They're mediators of intercellular communication because they contain nucleic acid, proteins, and lipids, which can modulate protein expression in the recipient cells. They can also pass the blood-brain barrier. And recent studies have indicated that eevees from Parkinson's patients' body fluids, such as CSF and blood serum, contain variants of alpha-synuclein. For these reasons, we decided to study how eevees from Parkinson's patients' CSF could propagate the aggregation of alpha-synuclein in healthy cells, in healthy mice, by intranasal administration, and initiate Parkinson-like symptoms and pathology. So we had two types of CSF samples. The first sample is from Parkinson's disease patients. The other sample was from a neurological controlled age-matched patients, which have neurological disorders, but they do not have aggregation of alpha-synuclein or Lewy body pathology. We will call them non-syncytiopathies patients. We began by using ultracentrifugation methods to isolate the eevees. We characterized them and observed their aggregation abilities in vitro. So first, we characterize our eevees when we use conventional methods that are appropriate for eevees research. We examined them for their morphology by TEM. We tested they have eevees markers expression like CD81 and CD9 in flow cytometry. And we evaluated their size and concentration by nanoparticle tracking analysis. Furthermore, we examined their alpha-synuclein cargo. We observed that both groups had alpha-synuclein variants within their eevees. Parkinson's patients had more pathological variants in them. We saw that they had phosphorylated 129 serine alpha-synuclein, which indicates a pathological conformation. And also they had aggregated alpha-synuclein. We also observed an expression of CD11b and CD113, which suggests that at least in part, some of these eevees are from a glial source. So first, we tested the eevees' ability to induce insoluble alpha-synuclein aggregates in neuroblastoma cell line. And we saw that only Parkinson's patients' eevees led to aggregations of alpha-synuclein, which was also phosphorylated in serine 129. And they were also positive for theoflavin S, which is a fibril mature marker. And of course, we saw this only when we looked at the Parkinson's patients' eevees and not in the neurological controls. And it also led to reduce the ability in these cells. So after we finished characterizing the eevees' ability to induce alpha-synuclein aggregation in cells, we moved on to our in vivo studies. So we treated young, healthy mice by a short-term treatment. This means only a few days and a very small amount of eevees from the patients. And we waited a few months, and then we tested them for behavior and pathology, which was relevant for Parkinson's disease. So first of all, after 3 months, which is a relatively short time, which we see in other models of Parkinson's disease patients, Parkinson's disease mice models, we first already saw that they had decreased motor abilities in the rotarod. And we also saw that they had higher anxiety levels in the open field test. We saw that they had decreased sense of smell. And maybe you remember in the beginning, I said that one of the first symptoms that we see even before the motor symptoms start in patients is actually olfactory dysfunction. So this was very interesting to see that these mice had decreased sense of smell. And we saw that in the hidden food pellet test. We saw that they gained light weight during the whole time of the experiment. We're not exactly sure if it was because they ate less, because they didn't smell their food, but all in all, this is a general well-being of the mice. And possibly my favorite trial and the least invasive test we did in the lab was we saw that they had premature gray fur. And this is very interesting. After 8 months, the Parkinson's patients treated eevees, mice just had much more white hair. And so after 8 months, we sacrificed these mice and we looked into the pathology inside their brains. And like I stated before, the hallmark of Parkinson's is Lewy body pathology, specifically in the substantia nigra. So we looked in the midbrain areas, the midbrain sections of these mice to better understand if we can actually see something that will explain the impairments we saw earlier. And we stained the midbrains for alpha-synuclein, phosphorylated alpha-synuclein presence. And we saw, of course, like we hoped and accepted, that we saw a lot of aggregations of alpha-synuclein in the substantia nigra, only of the mice that received the Parkinson's disease patients' eevees. We were unexpectedly also observed another area called the red nucleus, which was had a robust amount of alpha-synuclein aggregations. And when we looked up in the literature, we found that the red nucleus is it's not mentioned much, but it has a complementary role in motor control and coordination. So it actually makes sense that if we have these alpha-synuclein aggregations there, it could also explain why these mice had impairments in the motor controls. And once we saw these aggregations, we wanted to see if these aggregations led to the degeneration of these alpha-synuclein, of these if we saw this degeneration of the dopaminergic neurons in the substantia nigra. So again, we stained this area and we used TH, tyrosine hydroxylase, which is a marker for dopamine, and new N cells. This actually, positive cells for both indicates these are the dopaminergic neurons in the substantia nigra. And when we counted them, we saw that there was a substantial decrease in these amount of cells. So which would explain why we saw the motor coordination, the motor problems that we saw in the beginning. So we wanted to understand also the role of these eevees in the midbrain. Do they even reach the brain, or is something happening in the process? So we dyed our eevees, starting again, the eevees from the patients. We used PKH26, we labeled them, and again, we gave the mice, healthy mice, for 24 hours. We gave them the treatment intranasal, and we waited for 24 hours, and then we looked again into these areas. And we saw that these PKH labeled eevees, they went through the olfactory bulb and arrived in the substantia nigra. And we can see them, you can see them here, the orange arrows. This is indicating of TH positive cells in the substantia nigra that have the eevees inside of them. We can also see that some of the eevees are not in dopaminergic neurons. So in general, we saw that eevees reach the substantia nigra after 24 hours, and we can locate them in various kinds of cells. So going back to these mice that we sacrificed 8 months after, we looked into their pathology, and we were actually surprised that the aggregation of alpha-synuclein, the phosphorylated alpha-synuclein, we could rarely detect it in TH positive cells. This means that they're not found in dopaminergic neurons, which is interesting because only neurons produce alpha-synuclein. Where we did see them was actually in GFAP and EBA1 positive cells. GFAP is astrocytes and EBA1 are glial cells. And this actually means that there is some kind of glial clearance relative to this pathology of the brain. So to further assess the pathogenic process in the mice brains, we also looked at their striatoms. We looked at different types of pathogenesis that we also see in humans. And first of all, we saw a decrease in TH. This is a decrease of dopamine. And we already know that there is a dopaminergic neurodegeneration, and it correlates with this decrease in dopamine in the striatoms of the brains. And also we saw LC3 ratios, which were impaired, which will indicate some kind of impaired autophagy processes. And this is also something that we see in the patients. When we looked a little bit more for neuroinflammation, we were actually very interestingly saw this unique GFAP isoform. It's the size of 50 kilodalton, and we only saw it in the mice's striatoms that were treated with Parkinson's patients' eevees. You can see it here on the left. It's only the smaller band. We can only see it in these mice. And furthermore, we also noticed that there was a general increase in astrocytes and also microglia in the striatoms, which imply that there is an increased inflammation processes in these brains. So to summarize, healthy mice treated with Parkinson's disease patients, CSF-derived eevees, displayed Parkinson-related symptoms such as reduced motor functions. They had alpha-synuclein aggregations in substantia nigra and red nucleus. They had dopaminergic neurodegeneration, also in their striatum and in their substantia nigra. They had altered autophagy and neuroinflammation processes. So taken together, we believe that eevees have a major role in the progression and pathology of Parkinson's disease, and they should be considered when developing novel therapies for neurological diseases. And to conclude, this is the first research that shows the ability of Parkinson's patients' CSF-derived eevees to induce Parkinson's disease-related motor and non-motor behavior. It is also the first time to shed light on the role of eevees in the propagation of alpha-synuclein through the olfactory bulb by intranasal administration. And I want to thank you to everyone who was taking part of this study, to my PI, Professor Offen, and all my lab mates, and to our collaborators, Professor Dejalti and Professor Morenhauer, who has contributed a lot to this process. Thank you.
Shai, beautiful. Thank you very much. We have time for one question, if anyone has one.
Sure. Can you hear me?
Yes.
Thank you very much. This is a very impressive work that you have done, quite a lot of work. I wonder, what is the conclusion? How do these vesicles that you have taken out from the CSF of patients with PD, how do they find their way from the nasal mucosa or wherever just into the substantia nigra and red nucleus, and not perhaps to other neurons that are associated with Parkinson's disease, as you have mentioned? What is the theory? How do they get there? And
Well,
also,
it's a very good question.
if you can tell us, was there a dose response curve? Is one vesicle sufficient, or do you need how many? If you give more, will it increase the effect or make it earlier?
Yes, so it's a very good question. And first of all, regarding general eevees, so we know that eevees, especially we use eevees in the lab, just the opposite. We use mesenchymal stem cell eevees as a therapeutic agent. And we know that they have this homing effect, their ability to track neuroinflammation and reach the tissue that they're involved in. So in this specific area, we believe that in part, some of these eevees are from neurons, from glia, from astrocytes, and they have the ability to pass the BBB. And I don't think they only enter the substantia nigra, but I think they enter a lot of cells all over. But we believe that inside the substantia nigra, they do have the process of being pathological. I mean, the alpha-synuclein inside of them and other processes inside the eevees affect these areas specific because they have a more susceptibility to it. Does that answer the question?
Well, first, if I have a minute, I would say that you started with a assumption that actually alpha-synuclein are toxic to the neurons. And this is a prevailing theory, but it's not more than a theory, as well as the fact that the assumption that alpha-synuclein moves from one cell to the other and become toxic there. These are two very important topics of debate.
There are many articles that take preformed fibrils of alpha-synuclein. This means that these are alpha-synuclein which is already pathogenic, and when you inject them to various mice in various locations, it is enough to already start some kind of a cascade that eventually will lead to neurodegeneration. So alpha-synuclein by itself has already been shown to have some kind of a role in it. But in our case, we're not just talking about alpha-synuclein inside the eevees, but eevees as a whole multifactorial environment.
Thank you. I would love to continue the discussion maybe during the break, but we don't want to, on the first lecture, already be off time. So thank you very much, Shai, for that really beautiful lecture. Okay, we're going to hear now from Vijaykumar, who's a postdoc with Professor Danny Segal, and he's going to talk about the connection between Gaucher and Parkinson's.
Thank you very much, Karen. I'm going to share my screen. Okay, just confirm it's visible to all. Hello?
Yeah, we see it, but you need to press the button for presentation mode.
Okay.
Almost there.
Perfect.
Now it's good. Great. Thank you.
Okay. So very good afternoon to all, and thank you very much, PPD, for providing this wonderful platform. My name is Vijaykumar, and I'm a postdoc in Professor Danny Segal's lab at the Tel Aviv University. And today I'm going to talk about how a small metabolite that accumulates in Gaucher disease could be a risk factor for the Parkinson disease. So let's straight away jump to the introduction part. So Gaucher disease is a genetic rare disorder that's characterized by the deficiency of lysosomal enzyme glucosyl cerebroside, and due to the deficiency, it accumulates the glucosylceramide in the lysosome of macrophages. So in the normal scenario wherein the glucosylcerebroside is functional, it metabolizes the glucosylceramide lipid into ceramide and glucose. But in the case of disease, the gene that encodes the glucosylcerebroside got mutated and becomes dysfunctional. And after the becoming dysfunctional, it will no longer be able to catabolize the glucosylceramide, and due to which it will accumulate in the lysosome, and there will be formation of Gaucher cells, and which is detrimental. The Gaucher disease is known as 3 types. Type 1, which is non-neuropathic, doesn't involve the central nervous system. However, type 2 and type 3 are neuropathic, that involves the central nervous system. The GBA mutation has been linked with the development of Parkinson disease. There are genetic studies that were performed on the PD patients demonstrating that GBA mutation have a strong risk factor for the Parkinson disease development. And there are statistical data that estimates that the Gaucher disease patient have a higher tendency to develop the Parkinson disease in the later stages of their life. So this is really surprising that how, and there is no clarity over it, that how a mutation in a metabolic enzyme could lead to the Parkinson disease, or in other words, how a Gaucher patient in later stages of their life can develop the Parkinson disease. Well, in past couple of years, is more than a decade, Dennis Eagle's lab is involved in studying the amyloid formation and protein misfolding diseases, including Alzheimer's disease, Parkinson, or type 2 diabetes, which is associated conventionally with the formation of amyloids. And by definition, amyloids are proteinaceous aggregate rich in cross-beta structure that shows it affinity towards the thioplavin-T, and show bireferences upon binding with the Congo red, and they are fibrillary aggregate, and that can be visualized in the transmission electron microscopy. And they have a very special pattern of arranging the cross-beta sheets, which can be revealed under the X-ray diffraction. And very recently, there is another chapter has been included in the proteinaceous or conventional proteinaceous amyloids, wherein it has been mostly contributed by another faculty in the department, Professor Vijaykumar, who has observed in many of his publications that in his research that in certain metabolic disorders like phenylketonuria or tyrosinemia, or they found that even the small amino acid can forms the long fibrillary aggregate and show the similar characteristic of conventional amyloids. So here we were wondering whether the glucosylceramide, which is accumulating in the Gaucher disease, can also fall in the same category and forms the fibrillary aggregate that essentially can induce the formation of proteinaceous amyloid of alpha-synuclein, which is involved in the Parkinson disease. So to that end, our intention is were to characterize the glucosyl accumulate and their impact on the aggregation of alpha-synuclein, and obviously that will help us to understand the link between Gaucher disease and Parkinson disease. So first of all, we wanted to see if the glucosylceramide aggregate at all in the aqueous buffer or not, and if it aggregated can show the amyloid-like characteristic or not. So the glucosylceramide was incubated at different concentration in an aqueous buffer at pH 7.4, and you can see that after the incubation, we can see increment in the TST intensity that reached to a plateau after 3 hours, and the impact is like concentration dependent. The same impact in the turbidity assay. This is again signifying the size growth of particles. And when the aggregated sample were stained with the Congo red, it showed the amyloid specific apple green bireferences. That signifies that indeed the aggregate the glucosylceramide after the aggregation forms could be amyloid like characteristic containing. And then we wanted to confirm whether the aggregate does contain the fibrillar morphology as the conventional amyloid does. And for that, we did a gem analysis of the aggregates, and we could see that indeed these aggregates are of fibrillar morphology. And further, we did AFM analysis to get a more insight into the fibrillar morphology. And we could see that these fibrils have a twisted morphology. That's an average diameter of 52 nanometer, and the pitch height was measured to be average 29 and a pitch of around 184 nanometer. So in all, the take of message from here is that the glucosylceramide in an aqueous buffer forms these twisted morphology, and so the biochemical characteristic of the conventional amylase. And now the question is whether the lipid can form the aggregate at the lysosomal pH, because glucosylceramide accumulate in the lysosome, and the cytoplasmic pH is different from the lysosome. So we incubated the glucosylceramide in acetate buffer of pH 4.5, and we could fortunate enough to find that the glucosylceramide similar to the pH 7.4 also forms the fibrillar aggregate and show the TST kinetics as we observed in the previous slide, and also the turbidity. So we could make out here that the glucosylceramide can form the aggregate at the neutral pH as well as the lysosomal pH. After having confirmed the fibrillar morphology and fibril formation and amyloid-like characteristic of the glucosylceramide, we were interested in to see how it affects the aggregation of alpha-synuclein. And for that, in the figure A and B, see that when we did what we did here is we incubated alpha-synuclein alone and alpha-synuclein with the glucosylceramide, and we could here see that the alpha-synuclein alone, after having a log lag phase of around 25 hours, got a high in TST intensity and reached to a plateau phase in 50 hours. And at the same time, glucosylceramide upon incubation have a faster aggregation kinetics and reached to a plateau shortly after the incubation. But when the glucosylceramide and alpha-synuclein both were incubated together, we could see that they have relatively a longer lag phase and ended up a significantly high THT fluorescence, which is indicating that upon co-incubation, it forms the higher amount of aggregate, which is again reproduced when it's incubated at a higher concentration of lipid. And then in the next experiment, what we did is we incubated the alpha-synuclein in preformed form of fibrils that prepared on pH 7.4 as well as pH 4.5. And you can see that there is a concentration dependent impact on the lag phase of the alpha-synuclein aggregation, and also like we could see the same impact as we could see when the alpha-synuclein was co-aggregated with the glucosylceramide. So preformed fibrils also here we could see that it enhances the total fibrillation of alpha-synuclein at both pH and increases the lag phase, be it the acidic pH or near a neutral pH. The lower column is 10 micrographs that is showing the individual aggregation of both the component alpha-synuclein and glucosylceramide, as well as showing the co-aggregation of both the component, and likewise preformed fibril aggregate and their impact on the alpha-synuclein aggregates. And so, and in the next experiment here, what we did is we wanted to track the structural transition in the alpha-synuclein during the aggregation, as well as the impact of the preformed fibrillar aggregate on the conformational transition of alpha-synuclein. So first we did incubate the alpha-synuclein alone and track the structural transition at taking out the sample at different time points. And you can see that at 0 time point, the alpha-synuclein, which is a disordered protein, showing the negative electricity around 200, and that did convert into the beta sheet at 10 hours, and after that is showing the beta sheet formation that is specific to the amyloid formation. But when we incubated the alpha-synuclein with 50% galactosylceramide preformed fibril, we could interestingly see that there is an intermediate conformation which is absent in when alpha-synuclein alone is present. So here we could make out that probably it is because of the presence of glucosylceramide would be stabilizing some intermediate conformation in the alpha synuclein. And if you're aware of amyloids, then it is now it is well established that this is the intermediary conformation, which is much more toxic than the fibrillar aggregate. And to confirm this, first we check the cell cytotoxicity of glucosylceramide fibril alone. And here you can see in the figure C, that is control is the untreated cells, and 1% DMS is taken as vehicle because it is used to dissolve the lipid. And then if we incubate the CC neuroblastoma cell lines at different concentration of preformed glucosylceramide, you can see there is a concentration dependent cytotoxic effect of these fibrils. And the other experiment what we did is we have taken out the samples at different time points and wanted to see how the presence of preformed fibrils induces the toxic effect of the alpha synuclein fibrils. And we can see here that the after the 2 hours, the sample that aliquoted 2 hour, 15 hour, and 25 hour has the higher, the more toxic impact other than the mature fibrillar aggregate or the aggregate performed at the end of the reaction. So here the take-off message from here is that probably it is the glucosylceramide aggregate that stabilize the intermediate conformation, as I mentioned, and they may induce the toxicity by stabilizing this intermediary or oligomeric or prefibrillar aggregate conformation. There is a long list of glycolipid aggregation diseases, and our next intention is to see whether it is the something special with the galactosylceramide or glucosylceramide, sorry, or other glycolipid can also form the similar kind of aggregate. And for that we have taken galactosylceramide, gangliosides, glucosylsphingolipid, and galactosylsphingolipid. Galactosylceramide and galactosylsphingolipid, they are specifically involved in the Krabbe disease. The galactosylceramide and galactosylsphingosine, which is others known as phycosine, has been known to interact with alpha synuclein, and galactosylceramide specifically also found in the neuronal inclusion with the alpha synuclein. So after when we incubated the galactosylceramide or galactosylsphingosine in the aqueous buffer, we could see that the galactosylceramide forms the similar kind of aggregates, and they all also the similar kind of biochemical or biophysical properties as the glucosylceramide showed. However, galactosylsphingosine have a kind of weird morphology, and we are still trying to make out how and to understand that. Okay. Before we go to the Q and round, I would like to thank all these people and APPD for the financial support, and special to mention that this work in our lab is started by Dr. Ashim Paul, and he has done most of the work, and he should be given credit. Thank you very much.
Wonderful. Thank you so much, Vijay. For time purposes, we're going to skip and move straight. If we want to do questions during the break, we'll do so. We just don't want to start being too late. So I appreciate your consideration on that. So I'm going to introduce now Dr. Hila Tamir, who's a postdoctoral fellow with Dr. Jason Friedman. And this is really going to be, I think, I'm fascinated by the work that you're doing, using the piano for manual dexterity training in patients with PD. Hila, please, the stage is yours.
Thank you, Sharon. Okay. Let me just share my screen. Thank you for having me here today. My name is Hila Tamir, and I'm a postdoctoral fellow at the Movement Sciences Lab at the Department of Physical Therapy here at Tel Aviv University, working with Jason Friedman. And my background is in music. I am a music cognition researcher and a composer. And I will present here today 2 related projects from our lab: an open label experiment checking the effect of piano training on dexterity in patients with Parkinson's disease, which is about to start in Shiba's Movement Disorder Clinic, and an artificial intelligence and machine learning-based system that will both automate piano teaching and individually optimize it for dexterity training in patients with Parkinson's disease. But before I get to the projects, I will start with some necessary background about music interventions, and I will then talk a little bit about our choice of using the piano. But first, let's define the difficulties that we are targeting. Deficits in hand and arm function may be present in people with Parkinson's disease from diagnosis with impairments such as micrography, resting tremor, and difficulties in manipulating objects with appropriate speed and dexterity that are often reported and often are the things that make people go and see their doctor. And in fact, most of the movement and posture disorders that are frequently associated with Parkinson's, such as freeze of gait and bradykinesia and rigidity and tremor, can all manifest themselves also in the upper limbs and hands. And of course, as the disease progresses, these deficits in hand function progress too and can lead to limitations in work and recreation and fundamental daily tasks such as dressing and eating, and they have a direct bearing on patients' independence. And so dexterity is an issue that has quite a significant impact on many patients' quality of life. And while PD is progressive, studies suggest that motor learning can slow or even temporarily reverse some of its motor symptoms, including dexterity deficits. And music-based interventions have been found as quite beneficial in the maintenance of motor performance. And I would like to briefly survey the main studies in this area. Probably the best starting point would be the extensive literature on rhythmic auditory stimulation, RAS or rhythmic cueing. And broadly speaking, the shared characteristic of these interventions is that an external repetitive auditory stimulus is provided to the participants. It could be a simple metronome or something more musically rich. And this stimulus acts as a scaffold, so to speak, for the performance of some motor actions, such as walking, tapping, foot stomping, etc. And most of the experiments I know focus on motor actions that are themselves repetitive, with the main one being walking. And what we see from these studies is that RAS is a very effective tool that may lead to improvement of various parameters of gait, shortened freeze episodes, and in the case of a prolonged intervention over several weeks, reduce falls. And these findings are usually explained as resulting from the fact that we use different networks in the brain for self-paced movement and for movement that is externally synchronized. There's also an overlap, of course, in the thalamus and the cortex, but in the first case, we use the basal ganglia, which is usually impaired in Parkinson's, and in the other case, we use the cerebellum, which is more preserved. However, it is interesting to note two things. First, that not all external cues are made equal, and that auditory stimuli seem to be very effective, more than other modalities in improving movement. And another interesting fact is that using musical stimuli for rhythmic cueing has led to improved effects compared with just a metronome, both in gait and finger tapping. And these facts suggest that the neurological explanation that I mentioned provides a partial account of how auditory events in general and music in particular can inform and support motor action. Another kind of musical intervention in Parkinson's is instrument playing. And most research about this involves active music therapy, which, as the name implies, is a form of therapy that is commonly performed in a group setting that involves various types of motor activities, including instrumental improvisation, hand clapping, singing, etc. And it has been shown to significantly improve motor abilities and emotional status And 6 weeks of active music therapy with synchronized hand clapping and foot stomping led to improve overall mobility in patients with Parkinson's disease. Another form of music-based intervention is in Parkinson's is dancing. There are actually quite a few studies about the effect of dancing in Parkinson's disease. For those of you who are interested, here are 2 recent reviews. And the types of dance studies vary from Irish dancing to ballet, but there seems to be a substantial number in 2 particular categories. One is somewhat expected, methods that were designed to specifically address Parkinson's, such as the Dance for Parkinson method. And the other category is tango. The following studies are particularly interesting, in my opinion, because the intervention itself is not motor, but the measured effect is. The first is a study that demonstrated that listening to music may have a positive effect on arm and finger movement in patients with Parkinson's. And the second one shows how training in mental singing while walking without actually singing may improve gait. And lastly, there are some studies about the efficacy of training with auditory feedback using music sonification, which is the transformation of movement parameters to sound in real time. So, for example, sonification helps improve handwriting in patients with Parkinson's disease compared to the same practice with non-sonifying musical background and with no sound. And training consisting of action observation plus sonification, in this particular case of walking, led to significant sustained reduction of freeze of gait severity and duration in patients with Parkinson's disease. So this was a short survey of the auditory and musical interventions that target movement in Parkinson's. And of course, playing the piano, being at its core music and movement activity, may share some characteristics and advantages with these studies. But I would like now to talk a little bit more about the qualities that give the piano some edge when it comes to dexterity compared to other musical interventions, even those based on music playing. So why piano? One significant advantage that is unique to the piano is that it is extremely beginner friendly. It is very easy to produce sound from the piano. In fact, it takes only 1 finger to produce any sound in its vast range. In addition, the piano is designed in a way that makes it extremely easy for a beginner to understand the pitch to spatial location mapping from low on the left-hand side to high on the right. And this is not necessarily the case in most other instruments. In fact, being a composer, I've studied and written to most of the standard acoustic instruments, and I cannot think of any instrument that really competes with piano in this matter. Another important advantage that the piano possesses is its fixed intonation. Correct intonation is something that requires considerable effort on many instruments and involves skills that are only partially motor, for example, musical hearing, and very often frustrates and challenges beginners until it is acquired. So, and with piano, this issue is non-existent as long as it's a tuned piano, of course. But another important advantage the piano possesses is that while it is very beginner friendly, it nevertheless provides a wide range of possibilities of graded manipulation of fingers and wrists that vary from basic to virtuosic. If one wants to make the level harder, there's almost always a way. And last but not least, I would actually like to suggest that the piano is, in effect, a sonifying system, like the sonification studies I mentioned earlier, and a good one at that. And that's the important part, because arguably any musical instrument is a sonification system by definition, since they transform movement to sound, but not all instruments represent an effective sonification system. And for our purposes, effective means a mapping that represents enough important movement parameters in sound in a very easily decipherable and intuitive way, and time sensitive. And the piano is great at all of these. Some of the advantages include the fixed intonation that we have already mentioned. For many instruments, the produced pitch, the intonation, is a combination not only of spatial finger location, but also of the amount of pressure, be it air pressure or bow pressure. And this is not the case with piano. A finger located in a given spatial location will always produce the same pitch. It is also polyphonic, namely, it's capable of producing several sounds in parallel, reflected by the simultaneous movement of several fingers. A key that is depressed immediately responds with sound, the fact that provides excellent time resolution for movement representation. And I've already mentioned that the pitch to space mapping is very intuitive, so it's easy to understand from the sound where the fingers are located. And lastly, both hands are represented in sound symmetrically, as opposed to many other instruments in which both hands may be involved in producing a single sound, so that some of the movements the performer makes are represented in sound only indirectly. Here, every finger has a direct representation in sound. So what we have here is an instrument that gives very accurate and intuitive auditory feedback on movement, both in terms of space and timing. And that makes him a great candidate for musical interventions that target hand movement, and that is, and it is worthwhile to check whether the piano training can affect dexterity in patients with Parkinson's. And this is the main agenda of both studies that I'm about to describe now. The aim of the first project is to study the effect of piano playing on manual dexterity in patients with Parkinson's disease. This is an open label experiment. I will not go into all the tests and inclusion and exclusion criteria in full, but there are some interesting details in the design that I will highlight. So let's start with a chart describing the protocol. The first line represents the first week. The middle line represents the middle weeks, 2 to 5, and the last line represents the last week. You can see that each week consists of 3 main ingredients. There are the tests, an extended battery of which is performed at the start and at the end of the experiment. There is the guided training, which is an hour-long piano lesson, and 3 sessions of independent practice. So altogether, we're talking about a 6-week training program. An interesting detail is that is reflected by the chart that you have seen is that most of the experiment takes place remotely, and this is a point that might be relevant for many interventions and studies, and has become even more so since the start of the pandemic. So I will soon expand a little bit more about the unique solutions that we have come up with in order to run this experiment remotely, yet keep the setup very simple to operate on the participant side. The participants will receive during the first meeting at the clinic a light and portable piano keyboard and a MIDI recording device for at-home use. First lesson takes place at the clinic, adjacent to the in-clinic tests. In the rest of the weeks, there are weekly hour-long guided piano lessons. Most of them will be remote. And I mentioned there are also sessions of independent practice. So as you can see, this is quite intensive for the participants. And the musical material consists of a mix of standard repertory and specially designed keyboard exercises or etudes, if you will, that target specific issues in dexterity in Parkinson's disease. These may include, for example, various manipulations of finger synchronization and differentiation, and so on. So let me expand a little bit now about the solutions we have come up with for the remote setup. our goal was, on the one hand, to enable remote teaching, recording and testing, and on the other hand, to keep the setup for the participants as minimalistic and technologically light as possible. So for the keyboard, we chose a model that is very light and has a carrying handle. We built a MIDI recording device, especially for this experiment, that once is plugged in, the device stays on for the entire 6 weeks and records all and only keyboard actions. It's important to stress that this is not an audio recorder, so it's non-intrusive and it's capable of recording uninterruptedly for the entire duration of the experiment. And the last challenge we faced was that of communication during the guided online sessions, and the experiment doesn't require participants to own a computer, neither to operate one. We constricted ourselves to the use of the participants' existing smartphones. The keyboard comes with a note stand that enables positioning a cell phone in front of the participant, but there was also a need for another viewing angle, one that would enable the participants to see the screen, yet would show a video of the keyboard and the hands of the participants on the keyboards, so the teacher could give feedback on the playing. And we wanted to avoid cumbersome setups or any assumptions about the home space surrounding the keyboard. So we designed and 3D printed a simple phone holder for the keyboard that sits on the keyboard itself. So it sits on 2 adjacent black keys and is placed at the very end of the keyboard on either side. And it's also very easy to transfer it from one side to the other. And so we managed to create an operating home setup for remote piano instruction that is extremely portable and easy to operate. Here are some pictures of the setup. You can see that the phone holder is sitting on the keyboard, and the second picture on the left is the MIDI recording device placed next to a Rubik's Cube for the scale. And as you can see, everything is very portable, very lightweight, and the only requirement for the participants is to plug it in and to leave it as such for the duration of the experiment. We also have some remote testing.
Hila, I need to ask you to wrap up, please, because you've reached your time.
Oh, wow.
It goes quickly.
Yes, it does. Okay, so let me just mention the second project very briefly.
Less than 1 minute.
Okay, the second project we're working on is taking these ideas another step forward, and we're building a smart machine learning based system that does the same thing but automatically. Namely, it trains people on the piano, but it optimizes not necessarily for piano playing, but rather for dexterity training. And here too, some of the features are we're using very rich input, combining audio, MIDI, and motion capture sensors. The system reacts in real time and can correct mistakes in real time. We're using some remote testing methods that the QDG is a method that has been shown to correlate with the UPDRS assessments and can very sensitively quantify changes in fine motor control in patients with Parkinson. And the system will provide exercises that gradually advance in their dexterity demands. And recent advances in artificial intelligence will allow our system to learn how to optimize the exercises in a way that would lead to the best expected improvements in dexterity for a particular user. And in addition to that, the models themselves will be constantly updated and improved by the data on the outcomes from multiple users, what is called meta-learning. And that's it. It's our hope that this system will combine both the technological and computational power of the recent advances in machine learning with the fun and engaging platform of piano playing. Thank you.
Wonderful. That was really a breath of fresh air, different than what we're used to listening to. So no pun intended. Okay, so we do have a break now until 3:06. I think it's important for everyone to stretch and move around and do whatever else you need to do for a couple of minutes. If anybody wants to chat among yourselves, you can have some open discussion, but no obligation because we do have a 5-minute break now. Thank you. Welcome back, everyone, after that short break. And it's a pleasure for me to introduce Veras, who is a student with Professor Mia Horowitz in a collaboration that the group has with Dr. Moshe Shikoparnis. Please, Vera, let's hear from you.
Okay. Can you see my presentation okay? Perfect. Okay. So hi, my name is Vera. I'm a PhD student in Professor Mia Horowitz's lab. And I'm going to tell you today a bit about my study about the possible interaction between two genes, GBA1 and LRRK2, using a Drosophila fly model. So biallelic mutations in the GBA1 gene cause Gaucher disease, the most common lysosomal storage disorder, due to decreased activity of the GBA1 protein glucocerebrosidase, or GCase. There's accumulation of its substrate, glucosylceramide, and this occurs mainly in monocyte-derived cells. Our lab showed in the past that decreased GCase activity results from its misfolding, from ER retention, activation of ER stress, and ER stress response, also known as the unfolded protein response. And in the last about 20 years, it became known that carriers of mutations in the GBA1 gene have a higher propensity to develop Parkinson's disease. And not only that, but also GBA1-associated Parkinson is considered more severe than idiopathic Parkinson. This is mainly due to an earlier age of onset and just a generally more severe disease course. Mutations in the LRRK2 gene are also a known cause for development of PD. This gene encodes a large protein, which has a kinase domain responsible for phosphorylating many different proteins. Part of them are proteins that participate in intracellular trafficking, such as the RAB proteins. And it is known that LRRK2-associated PD is considered less severe than GBA1-associated PD. Interestingly, the G2019S mutation in LRRK2 and the N370S mutation in GBA1 are two of the most prevalent PD-associated mutations among Ashkenazi Jews. So it was very interesting in the recent couple of years, free publications, free clinical studies highlighted the modifying effect of mutant LRRK2 and mutant GBA1, stating that the GBA1 LRRK2 associated PD is less severe than GBA1 associated PD. So basically, these observations indicated there might be a protective effect of mutant LRRK2 on mutant GBA1. And this is interplay we decided basically to test. And in order to do that, we decided to use the Drosophila fly model. And the reason is because the Drosophila is very useful in trying to test interactions between two genes on any level they might be happening on. So in our flies, in our model, the flies express the endogenous GBA1 and the endogenous LRRK2 normally in the background. As you can see, there are two orthologs in the Drosophila of the GBA1A and GBA1B. And on top of that, we express two transgenes. So first transgene is the mutant human GCase. We began all our experiments with using the L444P mutation, just because it is more severe, and we wanted to try and reach maximum effect, show effect from the beginning. And the second transgene is the kinase hyperphosphorylating human mutant LRRK2. And here we started with the I2020T mutation, just because that mutation we had when we first started our work. And also in the recent years, it became known that the rate of phosphorylation, hyperphosphorylation of these two mutations is very similar, probably because they are, after all, just one amino acid apart. So first thing we wanted to make sure was that our method of expression does not cause any overexpression, and by that cause any effect on its own. So what we did, we extracted RNA from fly heads and tested for levels of expression using qRT-PCR and compared the transgenic human GBA1 and the fly endogenous GBA1. And while we did see some variability here between the two ages that we tested, we did not see, there is no significant change between expression level of the fly endogenous GBA1 and the human GBA1 transgene. The second thing we wanted to make sure, like I've said, the endogenous GBA1 is the endogenous GCase fly GCase is normal in the background, and we wanted to make sure that our method of expression does not alter in any way its activity or perhaps cause substrate accumulation. So we prepared fly extracts and added the substrate C6 and BD glucosylceramide. You can see that between the samples, the level of glucosylceramide was the same. And using TLC separation, we tested for the level of substrate that we were able to get. And you can see here our control flies express only the endogenous enzyme, and there is no difference in its activity compared to flies that have one transgene or even both transgenes at once. Here, of course, is the negative control. CBE is the inhibitor of GCase. And here on the right, you can see that we were also not able to find in our transgenes, whether one or both, any substrate accumulation. So we were able to say that fly GCase activity does not change in transgenic flies expressing human GCase. They express their endogenous GBA encoded GCase normally, and they do not accumulate any glucosylceramide. We have shown in the past that expression of human mutant GBA1 in the fly heads, in the fly leads to UPR activation and to development of Parkinsonian signs. And the hallmarks of Parkinson we are able to test, we tested in the fly were death of the cholinergic cells, movement disabilities, neuroinflammation, and shorter survival. So basically all of our design was, all of our idea was to test flies that express mutant human GBA1 compared to flies that express both mutant human GBA1 and mutant human LRRK2. So first of all, we tested for UPR activation, and the unfolded protein response is a cellular stress response that occurs when there is ER stress due to the misfolded proteins retained in the ER. It occurs in 3 pathways, all of them leading to upregulation of stress-related genes. And here we tested, using qRT-PCR, we tested for 3 genes, BIP, or as I'll show you in the next slide, the fly homologue of BIP, which is HSC70, also splicetex BP1 and ATF4. And also using Western blotting, we tested for the phosphorylation of the EIF2 alpha protein. So like I said, here are the expression of genes that we tested for using qRT-PCR, the BIP homologue HSC70, splicetex BP1, and ATF4. And we were able to see by day 22, and sometimes even already by day 12, in flies that had only the mutant, human mutant GBA1, a very dramatic increase in the level of expression of all 3 genes. And then when we tested flies that had both mutations, that had both mutations in GBA1 and mutant LRRK2, we could see a very significant decrease in the level of expression of those genes, and we saw that in all 3 of them that we tested. The other marker, like I mentioned, was the phosphorylated EIF2 alpha, which is its phosphorylation leads to arrest of protein synthesis in the cell. So here we did it, like I said, using Western blotting, and whatever level of phosphorylation we saw in flies that had only the mutant human GBA1, we could see a significant decrease in the level of phosphorylation once we looked into flies that had both mutant GBA1 and mutant LRRK2. So in the end, we were able to say that UPR parameters, which increase in L444P expressing flies, significantly decrease in the presence of the I2020T mutation. The next homework we tested for was the death of dopaminergic cells. And we did that by Western blotting for a protein tyrosine hydroxylase, or TH. This protein is produced by dopaminergic cells, and a decrease in the levels of this protein is a fine marker for death of dopaminergic cells. So first of all, you can see the dramatic decrease of the TH levels in flies that had the human mutant GBA1. And next to it, you can see flies that had only mutant LRRK2 and how much higher the levels of TH are. When we looked into flies that carried both the GBA1 mutation and the LRRK2 mutation, we saw significant improvement compared to flies that had only the GBA1 mutation. Their levels of TH were much more similar to flies that carried the LRRK mutation alone. So TH levels significantly improved in L444P GBA1 expressing flies in the presence of mutant LRRK2. The second hallmark was movement disabilities, and here we tested for flies' ability to climb. We did that both in 25 degrees and also in 29. Here, the 29 degrees temperature is sort of a added stressor effect for the flies, and in both cases, it was we could see very beautifully how flies that had both mutations in the GBA1 and the LRRK2 gene retained through the 3 ages that we tested the higher climbing ability compared to flies that had only the human mutant GBA1. So locomotion of flies significantly improves in L444B GBA1 expressing flies in the presence of LRRK2 at both 25 and 29 degrees. Next, we wanted to test neuroinflammation. There's a lot of talk recently about the fact that in any neurodegenerative disease, we should expect to see some level of neuroinflammation. So we wanted to try and see that in our flies as well. And in flies, neuroinflammation occurs in 2 pathways, the toll pathway and the IMD pathway, and both of them lead to upregulation of nuclear inflammation genes. We specifically tested for 3 of them, MTK, ATTC, and SEC. This was done on fly heads using TRT-PCR. And you can see that by age 22, flies that had only the mutant human GBA1, we could see sometimes a 5-fold increase in the level of neuroinflammation, and you can see the significant decrease in the levels of those level of expression of those genes in flies that had both mutations. So inflammation parameters, which increase in L444P expressing flies, significantly decrease in the presence of the I2020T mutation. The last homework we looked into was the lifespan of the flies. So here we performed a survival assay that was carried out in 29 degrees and analyzed using Kaplan-Meier. And you can see that the flies that had the shortest worst lifespan were flies that had the mutant human GBA1 alone. And you can also very clearly see the significant increase in the longevity of flies once they are introduced also the LRRK2 mutation. So survival of flies expressing mutant GBA1 is significantly improved in the presence of the I2020T LARC2 mutation. So to sum up, we were able to show the modifying effect of the human I2020T mutant LARC2 on human L444P mutant GBA1 associated PD signs. And I've shown you in this presentation that we were able to see how much lower the UPR activation was, the lower rate of death of dopaminergic cells, the improved locomotor abilities, the lower level of neuroinflammation, and the much longer lifespan of those flies. So thank you very much for your attention. And I would also like to acknowledge the Aufzien Family Center for Aufzien Family Center Grant and Aufzien Family Center Fellowship.
Wonderful. Thank you very much. Does anybody have a question? We have time for one.
It's an excellent, this is Roy Alcalay. This is an excellent talk. And I just wanted to make sure that, so one of the questions that people raise about LARC2 and GBA is that maybe the LARC2 is the dominant mutation and the GBA is a bystander that anyways 90% of those who carry it never develop PD. But your findings actually show that if you have a GBA mutation, the LARC2 can actually protect. Is that correct? So you would assume that the penetrance of Parkinson's is even lower in people who carry both mutations.
Yeah, so that is what we assume, that once we have introduced the LARC2 as well, we can see improvement in all parameters that we look into.
Congratulations for the fascinating findings.
Thank you very much.
If I can follow up with another short question. Do you think that the LARC2 might be protective in general and far beyond the GBA?
It's a very good question. I think it's we will be able to answer that once we better understand the mechanism of the way LARC2 protects in this case. So we have some theories regarding that, and we're currently working on them. I hope we will be able to answer that.
Wonderful. Thank you very much. Okay, so our next speaker is Ifat Weiss, who's coming from Professor Uri Asheri's lab, and she's going to talk about a platform that she has established and is examining. Please, Ifat.
Yes, just a second. Do you see my presentation?
Yes, now we do. Thank you.
Amazing. Okay, so my name is Ifat. I'm a second-year master student from Professor Uri Asheri's lab and Dr. Ben Maoz's lab. And my projects aim to establish an in vitro platform for examining aggregation and propagation of alpha-synuclein aggregates using super-resolution tools. Parkinson's disease is a common neurodegenerative disease known for both motor and non-motor symptoms. It is characterized with degeneration of dopaminergic neurons in the substantia nigra and abnormal accumulation of misfolded protein called the alpha-synuclein. Alpha-synuclein is a small cytosolic protein that is believed to be related to modulation of neurotransmitters released. It can be found in a lot of different kinds of conformations, and some of them may be pathologic. Studies show that the misfolded alpha-synuclein can bind to other alpha-synuclein and accumulate to form possibly toxic assemblies like oligomers or fibrils or Lewy bodies, which are the biggest aggregates and can be found in PD patients' brain. A hypothesis about the Parkinson progression was proposed by Brack, which suggested that the pathologic alpha-synuclein can propagate along the brain in a prion-like manner. And based on autopsy cases, they developed a staging system that characterized the disease progression according to the spreading of the alpha-synuclein. All of that actually raises a few questions. One is, at what stage can we observe the alpha-synuclein aggregate and which one of them are toxic? Is it only the big Lewy bodies or maybe some of the smaller forms. Does the alpha-synuclein propagate? We believe it does, but it's not a consensus. And if it is propagating, then which of the alpha-synuclein aggregates is propagating? Is it the smaller oligomers or maybe only the bigger aggregates like the Lewy bodies? And finally, if it is propagating, then how is it propagating? Is it something that requires high proximity, like through nanotubes, or is it something that can happen between cells that are further, like through exosomal release and uptake? My project aims to explore this question using a neuron-to-neuron platform that we built to identify the alpha-synuclein progression. It basically contains two separate cell populations, one that will be the donor of the alpha-synuclein and another that will be the acceptor of the alpha-synuclein. With this platform, we aim to identify and characterize the alpha-synuclein aggregates, and to identify and characterize the alpha-synuclein propagation. And finally, we also want to examine whether this propagation can affect the neuronal functions. So this neuron-to-neuron platform is based on organ-on-a-chip technology and was designed by Dr. Rosanna Rauti from Dr. Ben Maoz lab. It is basically contains two chambers. One of them is the well itself, where we culture one population of the neurons. The other one is a chip that we 3D printed that contains the other population of the neurons. At the bottom of the chip, we have a membrane with pore in it, so substrate can go through it. And we put this chip inside the well, so we have the two chambers. All of this can be modified for our needs. For example, we can control the distance between the population of the neurons, and we made it usable in super resolution microscopy. So after we established the platform, we went and creating our donors of alpha-synuclein. For that, we created viruses with the plasmid that we received from Avi Ashkenazi's lab. This plasmid contain HCN promoter, so it will express only in neurons, and the infected neurons will express cytosolic EGFP, and due to the P2A, it will express separately the cytosolic alpha-synuclein that is fused with HA. With this plasmid, it allows us to identify transmission of the alpha-synuclein that is fused with the HA from cells that express the GFP to cells that don't express the GFP. So first we made sure that the donors are acting like we wanted to. We made sure they are expressing the HA, that they are expressing the GFP, and we made sure that the levels of the HA and the GFP are correlated with higher levels of the alpha-synuclein. All these validation were made from our project student Eliran. After that, we took our donor neurons to the super resolution because not all the aggregates can be seen properly in light microscope, and super resolution allows us to get a much higher resolution. Instead of the 200 nanometer the light microscope can get, we can get up to 10 nanometer resolution. So we specifically are using the D-Storm. It's a machine that uses a specific buffers that makes the fluorophoton on and off randomly while it takes many frames. So in each frame, different fluorophores are activated, and based on that, we can calculate in high precision the localization of each fluorophore. So basically, the output from the storm is a list of localizations of the fluorophores. Here, what you can see is neuron that is a control. It's stained for HA in red and alpha-synuclein in green. As you can see, there is some level of endogenous alpha-synuclein. And here you can see the donor neuron, which we infected with the virus. And we managed to see some structure that looks like aggregation, but we wanted to analyze it further. The analyzation was done by Ophir from Professor Uri Asheri's lab. She used a cluster algorithm that she modified that called dbScan, which basically takes the number of neighbors that we want in radius that we want, and it searches for clusters. So we looked for clusters and we got all sorts of interesting data that we are still analyzing. For example, we looked at the size and density of the clusters, and we noticed that the mean radius that we saw for most clusters and the donors are of around 43 nanometers. And after that, we wanted to compare the clusters in the soma to the cluster in the processes. So even though we didn't see anything significant between those two clusters, we did notice that the clusters in the processes has slightly more localization, which means that aggregate in the processes may have more alpha-synuclein than aggregates in the soma. After that, we wanted to look at the propagation. So we took our chip, we cultured on one chamber the donor neurons, on the other chamber the acceptor neurons, and then what we expected is that the donor neurons will express the green, which is the EGFP, and here it's red, the alpha-synuclein that is fused with HA. We stained it for HA, and we wanted the acceptors to express the alpha-synuclein that is fused with HA due to the propagation from the donor to the acceptor. So we first took it to the confocal. In green, it's the GFP, in red, it's the HA. And we managed to see in the infected neuron something that may look like aggregates, and in the donor, in the acceptors, which are cells that are not in green, we saw the HA signal, which is the red. Sorry, okay. But we wanted to get higher levels of aggregation and propagation, so we used preformed alpha-synuclein fibrils. We added it to our platform and took it to the super resolution. Now, we didn't stain it for like normative alpha-synuclein, we stained it for the phosphorylated alpha-synuclein, which may be a better representative to the pathologic alpha-synuclein. And also we managed to find structures that may look like, that may be aggregates, as you can see here. And we did a comparison between the donors without the fibrils and donors with the fibrils. With the donors with the fibrils, we saw higher expression of the phosphorylated alpha-synuclein and more clusters of it. Just a second, it's stuck. Okay. And after that, we went to look at the acceptors to see if there is any propagation. Those are acceptors that were in the chip, were treated with the fibrils and are stained for the phosphorylated alpha-synuclein. We again saw structure that looks like aggregates, but we still need to finish to analyze the data, so we'll have to get back to you with that. And finally, in parallel, we are assessing how the alpha-synuclein aggregates and propagation may affect the neuronal functionality. This is actually still an ongoing experiment, and what we do, we use a multi-electrode array, which is a plate that at the bottom of it has electrode, so it can record extracellular activity, and it can also stimulate the neurons. So what we did all kind of comparison. For example, we compared acceptors to donors. We took also donors that were treated with the fibrils that we added, and this is still ongoing. We didn't finish to analyze the data, but what we can get from it is, for example, how many times the neurons are firing, like how active they are, and also how all this treatment affect their maturation during, as we measured by the burst duration. So to summarize, we were able to develop a platform which enabled us to use super resolution microscopy with our model. We managed to identify aggregation in donors using phosphorylated alpha-synuclein with and without the fibrils. We also identified aggregation in the acceptors using the phosphorylated alpha-synuclein with the fibrils. And we are currently comparing extracellular activities in donors and acceptors, which may indicate differences in the functionality. And that's it. Thank you.
Wonderful. Thank you very much. Does anybody have any questions? We have a few minutes for questions, and also if there are any questions from a previous session.
if there is a minute, I would like to comment on the issue that was raised by Roy and by Nir regarding the protective effect of LRRK2 mutation. I think that this is a misconception here. We know that having what we call a mutation in LRRK2, maybe 2019 or any other, are more people who carry them are more likely to develop Parkinson's disease. This does not mean that this mutation causes Parkinson's disease, more than to say the opposite, that not carrying them is protective. So there are many different polymorphisms in many genes, including LRRK2, and that we call LRRK2, the 2019 mutation carriers, incriminate this is only because it's a smaller frequency. Had it been the most prevalent one, then we would say that not having the 2019 mutation is protective. And I think that this is the issue. I think that we should not consider LRRK2 2019 as causative more than to say that not having it is protective.
Thank you. Any other comments or questions? Okay, so we will continue to our, let me get in here, our last talk. Guy Shapira, who is a student with Professor Noam Shomron, and he's going to talk about THC microdosing as an early protective Parkinson's disease treatment. And I should add that Guy just previously was awarded a travel award to go to a real live conference. That's very exciting.
Thanks, Guy. Thanks, Karen. Although it is a conference in Russia, so it's not really clear if I will be able to attend at all. Either way, so this is my talk. I'm talking about the neuroprotective effects of THC microdose and its potential applications in Parkinson's disease, because this is originally not a Parkinson's related project, but we had an happy accident and we found a bunch of evidence that actually proposes that it does have a really substantial therapeutic utility. So THC is derived from the cannabis plant, which is an ancient medicinal plant. It has an entire spectrum of metabolites. Many of those are currently being studied for their potential therapeutic effects. But most of all, THC sort of uncovered the endogenous cannabinoid system, which is this whole system of receptors and biosynthetic metabolic enzymes that regulates the system. So we are not talking about the kind of THC intake we see in recreational purposes or even in standard medicinal purposes. We are talking about roughly 100 or 1,000 times less, which elicits an entirely different response, even contrary to the standard dose, and in healthy and young model animals, it does have like a deleterious effect, a long-lasting cognitive deficit. But for model animals that were treated with some sort of neurotoxin or were subjected to some sort of acute insult, the microdose actually prevents the kind of cognitive damage you would see. And it was just tested with induced neuroinflammation and a whole array of neurotoxins And it's not even constrained to just the brain. It also provides liver protection and cardio protection, which is really, we can't even explain it yet. So the study I'm following up is actually used old and non-pathological model mice. So healthy, but old mice. And just the small microdose provided a significant improvement in cognition across many cognitive domains. And it included also this influx of neuroprotective and antioxidative factors, and very, very excitingly, neurogenesis in places you might not have guessed. These are not brain regions that are actually undergoing neurogenesis. So what I did is to take brain samples and do a full transcriptomic profiling and compare in 2 different time points, 5 days and 5 weeks after the administration of the microdose. And at both time points, I've just seen many general neuroprotective and actually some sort of negation of like a bunch of genes that are, their expression is associated with neurodegeneration, and the trend was opposite. So this was not just a couple of genes, that was nearly all of them. And just to save time, I'm not talking about the entire list of genes, but just a couple of very central transcription factors, such as SGK, which sort of rose up to a level that is more similar to what we've seen in our control group of young and untreated mice, and it actually does confer neuroprotection, and some of it is very specifically catered towards Parkinson's pathology. Move on, we have IGF-2. Now, this is an interesting one because look at that. This is the old untreated mice. These are the young untreated mice. And you can see there is a decrease, which is well known and well documented. But after just days after the administration of the microdose, we see a rise in expression, which puts it a bit closer to the young controls, and some of the pre-existing research is just fantastic. I mean, just by injecting it to the dorsal hippocampus of old rats, we can see a huge improvement in memory and mental capacity. And we have a bunch of very interesting properties that are more related to neurodegeneration, ALS, Alzheimer's, but we're focusing on Parkinson's specifically. And for Parkinson's, we have seen this extraordinary sort of cell survival response that is very specific to midbrain dopaminergic neurons. So just by increasing the gene expression of either one, SGK and IGF separately, we see like substantial increase in the survival of these critical dopaminergic neurons. And this is not just it's pretty hard to convey the magnitude of, how many cells does SGK1 actually rescues. And I think this figure from the taken from the paper shown in the previous slide pretty much says it all. So it's not only can we see more dopaminergic neurons that survive due to co-administration of SGK, but also we can see the alpha-synuclein is really constrained, and the way it propagates in a normal normally is sort of canceled out just by the SGK. And these are MPTP model mice, Parkinsonism. And this is not all, because we know from recent, more recent single cell experiments that we have this whole slew of genes that has the potential to actually save or at least partly rescue some of the very critical dopaminergic neurons of the substantia nigra. And I've seen a whole bunch of them change their expression accordingly in our experiment. So cannabis is already in limited use, we can call it, in Parkinson's patients. It is used mostly for its alleviation of motor symptoms and pretty modest anti-neuroinflammatory effect. But remember, this is not a conventional cannabinoid treatment. We're using just one cannabinoid substance, and at a dose at which its effect is completely different. So I don't actually know how relevant these effects are, but I do know that with the effects that are exclusive to the microdose, which is the cell survival response, we can potentially rescue at least a substantial fraction of the dopaminergic neurons in the midbrain that sort of catalyze the progression of the disease. So we might or might not have the regular effect on the microglia, which are useful, but they're essentially just treating symptoms at the progressive and later stages of the disease. But we do have this sort of promise of a novel, completely novel disease-modifying treatment that could delay or maybe stabilize the disease at a good rate and get us to a point where it is more treatable and we don't have to resort to the later and then sort of more drastic treatments that does not bode well. And that's about it. Next on my agenda is testing this treatment in model mice of the preclinical stage of Parkinson's. We are intending to get samples and use single cell transcriptomics so that we can actually apply or sort of derive these effects and associate them with specific neuronal populations. So it would be less general and very to the point, because some of them are very specific. That's about it. Thank you for listening.
Okay, thank you very much. This paper is now open for discussion.
So, Guy, thank you very much. This is an interesting approach. You're talking about dopaminergic neurons, and unlike your last slide, we see the patients clinically after they have lost many more dopaminergic neurons than you have indicated. So it's not after losing 20, but rather after losing 60 or 70 that they present to us in the first place. But the main issue is that the dopaminergic problems that we see there are not the main problems, because we can overcome the dopaminergic loss by giving dopaminergic drugs. It's the involvement of other neuron populations which are important. So I encourage you, when you go on with your studies, to look not just at dopaminergic neurons, but also at other neurons, unless you have an a priori hypothesis that it's just the dopaminergic neurons that will be susceptible to CB2.
Thanks for your comment. Well, the reason I focus so much on the dopaminergic neurons in particular is because that's what sort of directed me to this hypothesis in the first place. When I've seen, like, I think 50 genes that had some relation or were previously uncovered as associated with the death of dopaminergic neurons in Parkinsonism. And you're absolutely correct. And this is, I mean, we will sample other populations also. I don't have much to say about it because we don't know a whole lot about them, how it will affect them, things like the microglia or other critical components of the disease. Thank you.
Thank you.
Maybe as a follow-up to Professor Cochins and also to the first presentation from Danny's lab, we have gone beyond the dopaminergic cells and also beyond the neurons. As have been shown, the glia is affected, the synuclein is seen also, especially in the cortical glia, and obviously in the autonomic nervous system. All those part of the neural system should be looked at when we are looking for an effective intervention.
Thanks for your comment, Nir. This microdose treatment, it has some sort of general benefits that we see across neurodegenerative pathologies. I mean, it already proved itself in other models and Alzheimer models. So I can't really take these benefits and apply them across the spectrum of neurodegenerative diseases. But it will be interesting to see, things like, does the usual benefits of standard cannabinoid treatment apply here? Can we see like the very desirable anti-inflammatory effect, even if we use the microdose? And also this effect lasts. I mean, even after 5 months after the administration, we can still see significant effects. So regardless of the small dose, this is actually pretty substantial. Thank you.
Any more questions? So we have reached the end of a really fascinating, almost 2 hours. I think the most interesting and important outcome is that we are about 2.5, close to 3 years since we've initiated the center, and see how many different labs have joined into the Parkinson research from very different angles, from music to exosomes to cannabis and genetic Parkinson. Also, what I think we all can appreciate is the link between the clinical observation, the basic science, using biological materials from patients in the lab research. And I think this by itself gives us a huge satisfaction that the center is doing its job, is increasing the awareness and increasing the interplay between clinical and the basic science in Tel Aviv University. So it's a good opportunity to thank you all for this fantastic achievement. Thank you, Karen, for leading this. Without you, nothing would have happened, really. And Rinat,
I could say the same about you.
and Rinat for taking care of those monthly seminars. I think it's not trivial at all.
My pleasure.
And now when the new war is at the back of Europe, I think it's a good time to hope that we are, that science will lead to better days. Thank you.
Goodbye, everyone. Have a wonderful weekend.
Thank you.